When you look at this family photo right here, and I should note this photo is courtesy of Awkward Family Photos, you can see we have a mother and a father and 3 children. And looking at the children, that you can see that they have similarities to both of their parents. They don't look exactly like their mom or exactly like their dad, but a little bit like both. How does that happen? Well, as you know, you get DNA or genes from each of your parents. We have, as humans, 46 chromosomes in all of our somatic or body cells. So in an ideal world, you get 23 chromosomes from your mother and 23 chromosomes from your father. So we get 23 chromosomes from the mother's egg, 23 in the father's sperm, 46 in you. In this case, we have 3 and 3, which equals 6. Now, we've talked about mitosis and how we create cells that are identical to one another, but this is a little bit different because we can't use mitosis to make cells that only have half the number of chromosomes. Enter meiosis. So meiosis is the process of forming these sex cells or gametes that have half the number of chromosomes. So we're going to look at that process in this video. This picture is just showing an overview of mitosis, which we have studied already. So remember that during mitosis, after interphase, when the DNA is replicated, the cell goes through prophase, metaphase, anaphase, telophase, and then eventually cytokinesis. You end up with 2 cells that are identical to each other and to the parent cell, each with a full set of chromosomes. So we say that the cells that are produced by mitosis are diploid, okay, or 2N, which means, that's what that 2N up here means. Each chromosome has its homologous pair. So in all of my body cells, I have a chromosome from my mother and a chromosome from my father that are pretty much the same. They have the same genes on them, but they may have different versions of the genes. So that's diploid cells. That's mitosis. We're not going to talk about mitosis today. Now we're going to look at meiosis. Here we have an overview of meiosis. So the major difference between meiosis and mitosis is that in meiosis, we're starting off with a diploid cell, but then we separate in the first division. So we have 2 divisions, meiosis I, meiosis II. So in that first division, you separate the homologous chromosomes, so sister chromatids stay together. And then in the second division, you separate those sister chromatids. So you can see in this starting cell, in this case we had 2 chromosomes, and in each of our 4 resulting cells, you have 1 chromosome. Mitosis we use to make somatic cells or body cells, where we can see all the chromosomes are in pairs. But meiosis we can use to make gametes or sex cells, where each chromosome is kind of on its own. So we call that haploid. Here we have an overview of the first division in meiosis, also known as Meiosis I, and has a lot of similarities to mitosis in that the chromosomes go through prophase, metaphase, anaphase, and telophase. But there are some key differences, particularly in prophase and metaphase, and a little bit in anaphase as well. So we're going to be focusing on Meiosis I because Meiosis II looks pretty much just like mitosis, but with half the number of chromosomes. So right away, when looking at Meiosis I, hopefully you notice that in prophase, you still have the centrioles and the spindle fibers, so the nuclear membranes breaking down, so that's all happening. Our chromosomes have condensed, right, from chromatin into chromosomes. But here's a key difference. The chromosomes, in this case, are in pairs. Okay? We call these homologous chromosomes. You see it right here, homologous chromosomes. So the fact that these chromosomes are in pairs is really important, and they pair up all of chromosome. There are 2 copies of chromosome 1, for example. Those pair up together. There are 2 copies of chromosome 13. Those pair up together, and so on. You might notice something else happening here. Right here and here in this chromosome pair, you can see that they're connected. Okay, they're forming what are called chiasmata, and we're going to look at what's actually happening there in more detail in just a moment because that's very important. Okay. Another thing you can see that's different from mitosis is that in metaphase, they're still lining up in the middle, right, the kind of metaphase plate of the cell, but Now they're lined up in pairs. So they're lining up with their homologous chromosome. Remember, in mitosis, they line up single file. And then in anaphase, the homologous chromosomes are being separated from one another, but the sister chromatids are still connected. So what happens if we look at this starting cell right here has 1, 2, 3, 4, 5, 6 chromosomes, okay? And after the first division, we have 3 chromosomes in each cell, because keep in mind that when sister chromatids are connected to one another, they still count as just one chromosome. So this cell has 3 chromosomes, and then this cell has 3 chromosomes. What I want to look at now is 2 really important events that are occurring during meiosis I that increase the genetic variability within the cells that are produced during meiosis. During prophase I, so the very beginning of meiosis, crossing over occurs. So crossing over is when remember the homologous chromosomes are lined up with one another. So these are homologous chromosomes. And we call that event when they line up together, it's referred to as synapsis, as you can see here. So what happens is the non-sister chromatids, so the chromatids from the opposite chromosome, opposite homologous chromosome, so not the ones that are identical to one another. So these would be sister chromatids. These are non-sister chromatids. They cross over, as we can see right here. So this is that chiasmata, which you can actually view on a microscope if you're looking at cells in meiosis in prophase I. And so that chiasmata forms, they cross over, and what happens is some of the information from the cells actually switches place. So now we can see with our 2 non-sister chromatids that this blue chromosome now it's exchanged some information, has some of the genetic information from the red chromosome, and vice versa. So when these chromosomes separate during meiosis I and when the chromatids separate during meiosis II, what you'll end up with is 4 unique chromosomes that are all a little bit different, because these 2 don't have the exact same information anymore. And crossing over can actually occur multiple times on a chromosome. So it can separate many different genes. So that's one way that we get different variation. The second source of variation in meiosis occurs during metaphase I. So remember, during metaphase I, they line up in pairs, and then during anaphase I, those homologous chromosomes get separated, and the sister chromatids are still together. So one of the things that happens during metaphase I is what's known as independent assortment. And here we have a very simple example, looking at the chromosomes, and you can see that some crossing over has occurred in prophase I. So you could have a maternal chromosome and a paternal chromosome on the same side, and then paternal, maternal, or 2 maternal chromosomes and 2 paternal chromosomes, right? Either one is perfectly okay. It's just, chromosomes flip-flopping spots. So this one and this one, that's the only difference. This gives us 4 unique possibilities. There's a nice little mathematical formula that we can use. That's 2 to the n, where n is the number of, or the haploid number of chromosomes. So in humans, it's 23, right? Because we have 23 pairs of chromosomes. So that means for humans, it's 2 to the 23rd, which equals 8,388,608. It's an 8, sorry. Combinations for how the chromosomes can line up during metaphase. Okay? So you have that, plus all of the crossing over that can occur during prophase I, and it's no wonder that all of the gametes that are produced during meiosis are unique. After meiosis I occurs, right, and you end up, meiosis I, let's add on, is also known as the reduction division, because that is when the number of chromosomes has been reduced. Right, so meiosis I is the reduction division. So we've gone, in this case, from 4 chromosomes to 2 chromosomes, or in humans, from 46 chromosomes in the starting cell to 23 in each of the 2 cells produced by meiosis. And then we go into meiosis II, which is the second division consisting of prophase, metaphase, anaphase, and telophase. And if you look at these stages in meiosis II, they are pretty much exactly the same as mitosis. Chromosomes are now lining up single file during metaphase, sister chromatids get separated during anaphase, and then during telophase, all the same stuff happens. The only difference between mitosis and meiosis II is that in meiosis II, the chromosomes have already been reduced, so you're dealing with a haploid, or half the number of chromosomes. I'll leave you with this overview of the entire process of meiosis, including the 4 stages of meiosis I, and then the 4 stages of meiosis II. Okay, so remember meiosis I, right here, is the reduction division. We get genetic variation through crossing over in prophase and independent assortment in metaphase I. And then after telophase I is over, we have half the number of chromosomes, or haploid condition, in each of the 2 cells. And then meiosis II, very similar to mitosis, separating the sister chromatids. So in the end, you have 1, 2, 3, 4 cells, each have half the number of chromosomes. So meiosis produces 4 haploid gametes that can then be used in sexual reproduction.